I'm really excited to be here today to meet with everybody and talk about sleep. But I'm a psychiatrist. I'm not a sleep specialist. So what you will hear about today is how some of our work has gotten into thinking about sleep. So the title of the talk is Sensing Psychosis, because what I'll try to get you a feel for is how we interpret information from our environments in a way that's more active, really sort of going in and actually taking data that's about a latent system that we have to interpret. And so I'll start by thinking about how do we assess that someone has a mental health condition. You know, when we see someone on the street or when someone comes into the clinic, you know, there's this notion in the research world that you have cases, you have controls, you have people who have the condition, who don't have the condition, but of course, that's often going to be way more complicated than that. There's many different kinds of caseness, especially in psychiatry. We have many different disorders someone might have. There's also this notion that there's a continua from health to disease, and so we have to be able to assess where someone might be in that continuum. On top of that, we know there's this idea of comorbidity, where the same person might have multiple conditions that we have to be able to distinguish. And so it's a complex question. You can't just necessarily plot two biological variables together and expect to be able to discern, you know, who's got the disease and who doesn't. On top of that, we know that there's variance at multiple levels of analysis. We know that a lot of these conditions are heritable, so there is genetic variance. We've heard about that already. There's also going to be variance at the syndromic level, but we can't go directly there, right? We have to be able to, you know, the genetic variance is going to be causing variance at the level of cell assemblies and cell structures up to brain structures and brain networks, and then ultimately variance at the level of signs and symptoms, so the actual observable and subjective features that together come into these constellations that we call the syndromes. The trouble is that those variances are usually studied at the level of thousands of individuals. We don't bring a patient into our office and study all of these levels at once. On top of this, we've already heard this morning about developmental trajectories. So how do we assess that someone's mental health status is abnormal or normative depending on where they are in that developmental trajectory, right? So someone with schizophrenia, which is where my lab focuses, may have genetic problems that occur in utero, but those risk trajectories don't actually manifest as syndromes until that individual is up until their, you know, early adulthood. So how do we assess at any given time whether what they're experiencing is normal? On top of that, we know that there's changes in mental health that occur over a much finer time scale. So within bipolar disorder, which my lab also studies, you have periods where someone may be manic, and then they might be euthymic or relatively normal for a while, and then they might go into a deep depressive episode. So if you're trying to study the biology or make sense of whether someone's brain is normal, how do you factor in these sort of episodic state variances? So where my lab tries to focus in is really in thinking about control systems. So I know there's a lot of engineers in the audience, and so I think we'll talk a lot about this idea of working with latent construct models, right? So a latent construct model is one where you have some sort of sensory information coming in, but that sensory information usually is processed for features, right? It's not just raw sensory information, but there's some features that are most optimally mapping onto that latent construct that you need to measure, right? But then you don't stop there. You use motors and actuators to generate new information about that system, which we can call feedback. But that iterative process of taking some information from the environment and processing it through something that's been carefully honed is what we think about as this kind of working, you know, a latent construct control system model. And so the brain is basically a bunch of these latent construct models, right? It's taking information in from sensory systems, but those sensory, the brain networks are these feature extraction tools that have evolved over millions of years to pull just that key information that the organism needs to survive. And then it uses its own movements, you know, generates movements of the body to generate new information that it can then interrogate that environment. Clinicians are also doing this constantly, right? We can't just directly measure what condition somebody has in front of us. So we use our senses, we use our sight, sound, smell, et cetera, but we feature extract just the parameters that our training has helped us to figure out, right? We don't just take in the raw information. And then we use our own movements, our own nonverbal behavior, verbal behavior to gradually hypothesis test and zero in on what is it that patient has, what state are they in. But again, it's kind of it's sporadic, right? So not every clinician is doing this to the same amount because it is based just on their training. And so then what I'll tell you about today is how our group has started to put actual sensors onto patients in the environment. So sensors you can get off the shelf, like wearables, phones, microphones, video recording, et cetera. and use that same kind of iterative process to try to understand what mental state a patient that we're following might be in at a given time. And so nowadays, there are all of these new types of data sources that we can start to pull from. It's not just the old-fashioned, subjective reports from surveys, and we can even go beyond. We'll talk about it a little bit, but electronic health records and MRI scans and blood and so on. But all of these other data types like location and movement patterns, voice and video that you've already heard a little bit about. But just as we were talking about, it's not just those raw signals, but we want to really understand how to optimally feature extract all of these signals as we think of them as these sort of more emerging medical data types. But ultimately, again, for the focus of my talk, we'll be thinking about how we map those kinds of values, those kinds of features into relevant psychiatric variables. So we'll talk today about sleep and circadian, but also things like executive function, language, depression, and so on. And so just to kind of zoom out a little bit more, the institute that I co-direct at McLean Hospital sort of has this bird's eye view of where the field of psychiatry, and this probably could extend to other branches of neurology and medicine, but is really to think about comprehensive phenotyping up at the top as being something where you're now pulling in all these data sources and aligning them together with data from the medical record, and then using that to design systems that you can interact with people at the center of care, whether that's applications or predictive analytics that you could pass on to payer organizations and so on. I'll be focusing mostly at the top here, but I will tell you a little bit about some of the work we're doing using sort of a human-centered design approach to provide these data back to clinicians and patients in a way that will motivate improved care and behavior. And so I want to kind of walk you through 3 use cases or scenarios of where we think that data science can be especially useful for psychiatry. The first one, which will map on, I think, a little closer to the previous talk, is work we're doing on inpatient sleep and activity assessment. So taking psychiatric patients and really begin to take a much more precision approach to their movement patterns while they're inpatient. And then I'll talk about something that's sort of where the rubber hits the road for psychiatry, which is dyadic interactions, right? It's that patient-doctor interaction, which is really where a lot of treatment occurs, but it's also where a lot of assessment is still going to occur. So we thought that it would be important not just to measure sleep and other things like that, but really to start to measure what happens during that dyadic interaction. And then finally, I'll tell you about how we're trying to pull this all together into more of a remote monitoring or sort of outpatient long-term assessment ecosystem that includes a lot of these components. So for the first part, we're really just going to be focusing on a couple data types. So wearable data from a watch, electronic health record data, and then feature extractors that we've generated, something called DP Sleep, which is a way to take raw accelerometry data and turn it into sleep-related values, not EEG level sleep. And then the electronic health record data we're using just to link medication administration with actigraphy. And as you'll see, the idea here is to actually generate user experiences for doctors initially, but also then patients to be able to deliver these insights back to clinicians. And so for the last few years, we've been using this research-grade wearable called the Gene Active Watch. It just records triaxial accelerometry, so it doesn't have the PEG sensor or any of that. But it is able to capture this at like 20 hertz for like a good 30 days without needing a battery refresh. So that, in the inpatient use case, is really great because we drop it on patients when they first get to the hospital. We don't have to take it off till they get discharged. And then at the day level, you can see something like this, which is, every minute of the day you get an activity level, and then you can derive these activity classifications. In this case, it's using a percentage value, percentage based on that person's overall level of activity. So if you're not very much of a mover, it's going to adjust accordingly. And then from that, we can derive these periods of wake and sleep, and then, as I'll tell you about, we can featureize those periods to give you some statistics about it. And so then you can generate visualizations like this, like over an entire hospitalization. You can see this person at the beginning of the hospitalization really wasn't sleeping at all. So this was probably someone who came in with a mania. And then their sleep normalized, and then you can see towards the end of the hospitalization, they had some periods of really low activity and some longer sleep epochs. And so we feel that just helping to measure this, even though it may not be as accurate as like a polysomnography, it really helps guide clinicians just to have access to this in a more real-time way. And so what we've been able to do is then, like we heard in the previous talk, start to generate distributions for all the different, sleep parameters we can derive in this setting. We also can link this to specific medications the patients have received during their hospitalization. A lot of patients come in with psychiatric hospitalizations, having issues with sleep, and so there's a lot of lore, frankly, about, was trazodone work well, does Seroquel? The doctors, don't necessarily measure this, so we feel that this is helpful for them. And then where we've really been focusing is generating these activity reports. So again, trying to derive something that's relatively easy for someone to understand. So it's kind of in that explainable AI area, but still something that they can use. So the idea here is, what we do is put some sort of large values at the top for the clinician, something that summarizes the overall duration of sleep, the quality of sleep in terms of fragmentation of sleep. We are very interested in waking activity, as I'll get into more. When patients with depression are awake, they're also not moving very much, right? Even though if they get a lot of sleep, they're sluggish. Patients with bipolar disorder, they're often pacing and moving a lot, right? So we really see this as a holistic view of the 24-hour cycle, not just the sleep part, but really the full pattern of that person's sleep and wake activity. We also, the idea is that each medication that this person's being given of the day of the hospital is color-coded, so that, and the doctors actually gave us the feedback of what they wanted to see and what colors and all those kinds of things. So this way they can quickly see over an entire admission which drugs were started, which drugs went up, which went down. We also show these colored dots, so if the doctor is really interested, they can see exactly what time a drug was given relative to when the sleep epic happened and so on. So that's what's being shown here. And as we heard earlier, sleep is a behavior. We're very interested in using actigraphy to look at a range of behaviors. So sleep being the one that's sort of the most obvious to recognize with the accelerometer-based activity classification, but there's a lot of work that's being done. Brushing teeth is something which is relatively easy to measure with an accelerometer. And you don't think of it as that relevant, but when your activities of daily living start to fall off, sleep changes, but so does things like brushing teeth, taking showers, and so on. So using accelerometry to capture the activities of daily living, which again are not just common to all humans, but are something that like all animals have, to some extent have these behaviors. So we'll come back to talking about this, but I want to shift gears now and talk about these capturing dyadic interactions, and we'll come back to sleep in a bit. This part of the talk will focus more on linking up subjective data with data from video and audio, and then a little bit of how we then visualize that more in a research context. So we started this because when we think about that kind of multi-level research enterprise, we're trying to link genetic variants or biological variants with syndromic variants. The syndromic variants part, you sort of take for granted. Oh, you interview them, you get the diagnosis, and you're done. But the reality is, that's the most expensive part and probably the noisiest part of our whole research operation, because it takes months, if not years, to train people to do these kind of assessments well. And yet, even with that, the administration of this and the scoring takes a long time, and it's still highly subjective. And this doesn't really work well for longitudinal studies where you want to follow someone's clinical state and their brain state as it's changing over time. And another way of putting this is it's just very human resource intensive and it's expensive, and yet it's unreliable. So how do we do something that's more reliable at capturing those dyadic interactions? So we partnered with some computer scientists. We, at the time, didn't know how. We knew that we should start recording our interactions, but then what do you do with that data? And this was 10, 15 years ago. So we went on YouTube and we found this group working out of University of Southern California doing really cool, computer vision. And basically in real time, they can get all of the facial expressions, all of the pose data, all of the ways that someone's speaking just from a regular camera and microphone. And so what we did was to try if we could collapse all of the information that we're capturing over hours into just a few minutes. We took as our kind of clinical situation, the morning rounds encounter. Because every day on a clinical unit, the doctor has to figure out what somebody has and what treatments to make within the span of a few minutes, right? They have just a few minutes to talk to that person and do their full mental status exam, and on that basis, get all the billing done and get all this person's life-changing decisions. So we took clinicians from the unit and again helped them co-design the semi-structured interview that they could do in a way that wasn't awkward to do every single day. And so with questions like, can you remind me what brought you to the hospital, sort of open-ended question, followed by these other more pointed questions. Has anything been on your mind lately? What are your goals for the hospitalization? How are people treating you? These people don't ask, are you feeling paranoid? They say, how are you people treating you, right? So these are more naturalistic probes, the motors and actuators that clinician is using to derive the feedback that it needs, or he or she needs to make a clinical decision. So what you can see here is that the participant is visualized at the top, and evaluators down at the bottom. I think of this sort of like the behavioral EEG. You have the different rows here representing facial action units. And you see while the participant's alone here, you can see them looking around, raising their brow a lot with the red colors here. As soon as the doctor enters the room, they generate a smile. And then in response to each of the probes, the questions, you can see their facial affect changing. You can also see the evaluator, in this case, not smiling much, but doing a lot of squinting and raising their brow. So within a very brief encounter, you can start to really see the richness of what's going on in this dyadic encounter in a way that's still privacy preserving. We don't know who's in this encounter. We can't actually interpret this, but it is something that we can then begin to interrogate in different ways. So we can then look at the responses, the facial action responses to each of those probes, so aligning all of the questions together. And so, for instance, patients who would go on to get rated as being very highly depressed actually showed a decrease in the variance of their brow raisers or their brow furrowers. So it's not that people who are depressed frown a lot, but they do show reduced variance of their facial action, right? So they're not actually, and the more depressed you were, the less variance they saw. And then, interestingly, in the alone period, which was there on purpose, we know that patients with a psychotic disorder, when they're by themselves, often do like self-dialoguing, or we call it responding to internal stimuli. So in fact, when patients were alone, those who would later be evaluated as having unusual thought content did a lot of social behaviors. They were smiling, they were doing the brow raising. So the idea here is that you can get these signals and you can couch them in the correct context, just like a clinician would. And so a lot of these things are basically taking what we call clinical pearls, things that clinicians do and use every day, and then trying to see if we can derive something that's basically a filter for that pearl. And then this is just showing that you can then use these kinds of tools to predict conventional scores, like, this is just a simple leave one out model where we can get the negative symptom inventory, which is one of the dimensions of psychiatric symptoms we're interested in. And this is just visually, for those in the audience who are familiar with like a spring-loaded graph, these are the conventional items. The PANS, the MADRS, and the BPRS are clinical trial targets. These are things that we would use to judge someone's responding to an antipsychotic or an antidepressant. These are all the individual items of those individual scores or instruments. And then we can just begin to superimpose the values that we can derive from our automated system, right? And so this is a very early proof of concept that we did some years ago. The idea is to collect more and more data in more and more context to basically establish criterion validity for whichever of those constructs are meaningful or important for the clinician. And as I mentioned, we also are very interested in data visualization. And so one of the goals that I had as a PI and also just thinking as a psychiatrist, what would I want to see about an interview that someone else did? If I didn't have time to sit there and watch the whole interview, how do I know it went well? What can I see about this person's body language and so on? And so this basically is a real-time report that we can generate within an hour or two of the interview that gives us a lot of information about the participants' body language. So this is like their gaze and their pose data, their facial action units, as well as those of the interviewer. And then we get a similar kind of report about the audio data. And then down at the bottom is sort of a summary of the usage of words that were done in that encounter. So speech rate, speech coherence, word uncommonness, and then a word cloud of what was spoken. And the idea, again, here is that we really want to be able to see not only the time series, but we also want to see the correlation. So we really do explicitly model all of those facial action units relative to one another, because they don't just occur in isolation, right? When you have a full smile, certain action units come together. When you have a fake smile, only part of those action units come together. So we can spot fake smiles versus real smiles based on the correlation pattern we get here. And then we can see how that pattern deviates from a group level dendrogram, which is shown here. And so in this visualization, above and below the diagonal, I can see what this participant did in this particular interview and how they looked compared to themselves overall. So it's sort of that person relative to themselves and that person relative to the group. Okay, so in the last third of the talk, I want to talk about how we're trying to pull this together into a remote monitoring situation. So, when we think about patients who have a bipolar disorder or schizophrenia, what does that even mean? These are kids in their 20s. They come to us at the time when they are maybe are struggling with something. But like, who is struggling with something, right? Especially at that age. So, we think about this as, kids driving down Route 1. I was just on Route 1 in San Francisco earlier this week. It's beautiful, but it's dangerous, right? That's what kids want to do. They have a changing attitude towards, what kind of behavior makes sense to them, and that's developmentally normal. But how do you then superimpose on that the actual experience of schizophrenia or bipolar disorder? Because these kids will tell you that once they do have an episode or they have to go inpatient or whatever, it doesn't just take weeks to recover, it takes years to recover. And so really observing that in relation to their life was something that we thought would be important, because until then, a lot of the studies were just done at the level of thousands of people scanned, or assessed at one point in time. And so we were fortunate to be part of an NIH initiative called the Intensive Longitudinal Health Behavior Network. So this was actually across multiple of the NIH institutes. So sedentary, there was some in National Cancer, there was NINDS, there was a number of them. Our project was focused on following 100 individuals with schizophrenia, bipolar disorder, or major depression, follow them for at least 1 year with the idea that that's about the time span that they're likely to experience significant fluctuations, and then be able to track subjective reports with a phone app, clinician evaluations where they would come in for recorded encounters, and then objective measures of sleep and activity from watches and other sensors. And so this is sort of the timeline of an individual participant. So each participant wore that same Gene Active watch I mentioned. They had an app called BWI, which was a Harvard School of Public Health app. They then came in for study visits the same way you might in a regular clinic, like every month or so. And then they also performed web-based neuropsychological battery at each time point using something called Test My Brain. So from those raw data sources, we can pull a number of features. So there's these passive data features from the watch, including active, like button presses. We can get a number of features from the phone, as well as audio diaries and surveys from the phone. And then when they come in or go, so pre-COVID, they came in physically to see us, and then they had Zoom-based encounters where we can score all of those conventional clinical trial measures, as well as capture the information from the encounter. And from that, I'm not going to go into it too much today, but since we're talking about sleep, we were very interested in this issue that the latent sleep, the latent energy that patients were experiencing. So they clearly patients who had depression seemed to be less energetic than other people. People with bipolar disorder or mania seemed to have way more energy than other people, but it seemed to be something like maybe there was a misalignment between actual energy and perceived energy. It was sort of the way we were coming at this. So we really wanted to measure a lot of those kind of those discrepancies between the actual and perceived levels of energy. So I want to walk you through one participant who was with the study for over 700 days. So what you can see in her is, what I'm showing is her responses to a bunch of EMA questions. So these are phone-based surveys that she answered each day. And basically, when she started the study, she was reporting more being energetic, alert, inspired. And that occurs for the first 100 days or so. And then right around then, you can see that she shifts into feeling more hostile, more irritable, more ashamed, and so on. And then that period also lasts for almost 100 days. And then again, she shifts. And you can see this really striking, almost oscillatory behavior where she's going for these long episodes, and then towards the end of the study, the second half of the study, she's still going through episodes, but they're more rapid. So this is someone who has really profound and dense depressions, and then you can really see that. And when we align that with the conventional scores, it's what you would expect. She does get rated as having higher depression when she came in during a period she used to be depressed. But look what happens towards the end of the period. We don't see that variation anymore because she's only coming into the clinic when she's feeling better. She doesn't feel like coming in when she's doing worse. By the time she gets in, she's doing better, but she's actually scored as being high because the way you score these things is over the past 30 days, how depressed were you, right? And so it creates a problem when you're coming in and someone's getting scanned, but it's only because they are actually doing well enough to feel better, right? And so then I can also show you where it gets interesting is what her objective sleep data starts to do. So what you see is that in the beginning of the study, this is her sleep pattern. And right when she starts to feel depressed, what happens is she starts to wake up much later. So she still goes to sleep at around the same time. But notice that her sleep gets later, or her waking time gets later. And then also notice what happens during the day. So the really dark, or the brighter red color here, shows that she's very active during the day. But during that period of depression, she's much less active during the day. So just what we were saying before, someone who's sleeping a lot, and yet even during their waking period, they just feel sluggish. They're not moving around much. And then when she starts feeling well again, that shifts right back. Her sleep starts to be shorter, and she starts to be more awake again, or more active during the day again. And so what we thought this would be an interesting way of modeling this is to actually think about this as almost like a battery, okay? So thinking about this as like someone who needs to charge their battery every day, but if you don't get enough sleep for the amount of activity that you had during the day, it's just you didn't charge the battery quite as much, right? And so what would be the consequence if someone were to like be not charging their battery enough over a long enough period of time? Might they eventually get into a situation where they had to shift into a more conservative energy posture to really recharge the battery, right? To kind of go through a period of time where it was a much more bias towards resting, right? Same way if you had a virus, right? If you have a virus, your body is telling you not to move as much so that you can recover faster and use those sleep processes to manage all of that. And so we modeled this as basically a simple homeostatic system where for every minute that you're awake, you're spending a little energy, and the more you move, the faster you're spending the energy. And then while you're asleep, you're recovering the energy. And the more you're immobilized, the faster you recover the energy. That's the only assumptions the model makes. It's not based on any sleep biology per se, although we're certainly influenced by thinking about sleep pressure and so on. But what you'll notice then, if you just make that assumption, that the time that you see this inflection point in her energy balance is right at the same moment in terms of the days that she's actually having those subjective changes. And so we're really interested in this idea that perhaps depression and potentially mania are like energy conservative processes that are there to help the system stay in a kind of homeostatic balance. And so we're in the process of applying that hypothesis to a lot of other data that some of which I'll get into. So one of the ways we wanted to look at this is with UK Biobank data. So in that case, we have 1 week of actigraphy from 100,000 people. So it's not, and they have a ton of other deep phenotyping that they do for that week. So it's not as longitudinal, although there have been longitudinal follow-ups. And so we used our DP sleep algorithm, the same one that I showed you in the previous slides, and we just generated a discovery data set and a replication data set of about 38,500 people. We then ran our algorithm and did some manual QC to create usable data sets. And then on the right is basically the average week of a human sleep-wake pattern, right? So you see that in both, basically in everyone, the weekends, you're waking up a little bit later, right? And then you actually tend to be a little bit more active, and that's very consistent, right? And so then that's based on the whole data set. But what gets interesting then is when you look over the lifespan, you see that as individuals age, their daytime waking activity goes down quite a bit, and then you can see how the changes in the week versus weekend, that's so-called social jet lag, that also becomes much less marked as individuals age and they stop working. But then we can do really simple things like compare people with and without depression. So here in red is people who endorse depression in that UK Biobank data set. And so you can see how that affects sleep onset time, waking time, sleep duration, and waking activity as individuals age. And so I want to draw your attention to the one on the bottom right here, waking activity. So notice just how linear that is. So in both males and females, as you age, your amount of waking activity is dropping in a very linear kind of way. And what's then remarkable is that people who report depression have less activity, as if the whole curve has been shifted by about 5 years. So it's basically like whatever your age is now, if you were depressed, you would move as someone who's about 5 years older than you. At least that would, that's what the average shows. And so we thought that was like an interesting way to then start to assess patients in our study. And so we were already hearing about growth curves and developmental trajectories. So we did the same thing now, but with the older side of the population. So starting at age 18, where we have some data, you can see all the blue dots are the UK Biobank data set. So that's where they studied. They started at age 45 and up because they were interested in dementing illnesses and so on. We don't have a lot of data here, although there's probably data sets that I'm not aware of. But in terms of things that we can derive from publicly available data sets, we can then begin to derive these kind of growth curves about what's a normal level of waking activity. And then we can apply each of our participants in that longitudinal study to see where they fell. So that woman that I showed you a moment ago is this collection of dots here. And so what you can see in her is that when she was doing well, her activity was right in the middle of her age norm. And then as she became depressed, it dropped into that 20th, even down into the teens percentile. And the relationship in her of her mood and that differential was really striking. As if, in this person, you could even use it as a state-level biomarker. So then in the last few minutes, I want to talk about location. I think that's another really interesting contextual signal that gives us a lot of information about human mental health. So here's an individual who we followed for over 2 years, I think actually like 4 or 5 years, but this is the first 2 years of his participation. And this is just showing where he goes on a daily basis. So again, time of day is reflected on the y-axis, and then days of study are on the x-axis. And you can see the gray location is his home location. That's where he spends most of his time, of course. But then you'll see a number of these other kinds of locations that pop in. And so the blue location is a location that he goes quite a bit during the week, in the evenings especially, but also during the day. And then you'll notice that this is also a location that he goes for like 10 days straight here in these two parts of the study. Any guesses what that could be? What's that? Hospital, yeah. So this is the hospital. So this is a guy who has schizophrenia, bipolar disorder 1. He goes to therapy. Actually, you can see it even more clearly here. So you can see that he's going to the hospital for therapy on Wednesdays, but he also goes to the AA meetings at the same location on Wednesday and Thursday evenings, and you see also again some Saturday evenings. But then those longer periods were when he was hospitalized for mania. So you can very quickly derive where people are going in the context. This doesn't require much AI, right? This is just deriving what kind of location it is. But we think even without knowing the exact latitude and longitude, we can derive models that will determine what kind of locations these are based on some of these patterns. So for instance, this teal location here and this black location here, these are part-time jobs. You can really see that here, where Mondays and Fridays, he's first going to this part-time job. Later in the study, he's going to a different location on Thursdays and Fridays. You can also see that in the middle of the study here, after that second hospitalization, he doesn't have any job. He's really much going back and forth between his house and that hospital location. But then you can see around here, right before that second part-time job, he also starts going to this orange location a lot. It's somewhere that he not just goes during the day, but he also sleeping there. Any guesses what that one might be? What's that? Girlfriend, yeah. So this is a significant other, right? So you can then very quickly see community functioning, social functioning. You can see that the relationship didn't last super long, but it is remarkable that with just one sensor, you can pull out all of this rich community information about somebody. I don't think I have a slide to show this, but do you see these little red dots everywhere as well? So they're populating every day of the week, and they're often surrounded by his home location. Any guesses what that might be? Good question. Yeah, Starbucks. Other things you do outside that are walking dog. Yeah. So if I told you this guy's a smoker, So he can't smoke in his house. A lot of people don't smoke in their house. So he goes outside to like a specific set of locations to smoke. So then you can start to see that what we would be wanting to do is to link his actigraphy data, because we have him wearing his watch on his smoking hand, with the location data to start to actually use location and actigraphy as a way of sort of looking at the joint probability of when he's smoking. So, we think a lot of these techniques that are focused on like a single sensor have a much harder time. Doing gesture recognition of smoking is like one of the holy grails of actigraphy, but linking that with patterns of where people go and thinking about location as a way of contextualizing all the other signals you can get and really making the problem a lot easier, right? Sort of like a topic modeling for sensor data. This is just showing 7 examples now of other people in the study. And the point I want to give here is that, just having walked through it for a minute or two, you can start to see how each of these different people's lives might be quite different from one another. Just based on, this person is moving in and out of dorms in this case, this person has a very simple life and so on. So it's a way of getting at community and social functioning in a very low burden way. Just coming up on the end of the talk, I want to emphasize that this does bring up a lot of ethical, social, legal questions about when and where these technologies could be useful. Before this, our lab was really focused on neuroimaging, where we wanted our work to be relevant, but here we're starting to see that it's, has these use cases that we never intended it for, but there really could be places that these tools could get used. So as a part of that work, we generated something called the ethical checklist for digital health research. So if you're doing this kind of work or interested, or not familiar with this, and you want to do it, this really takes you through all of the elements of sort of how informed consent is different, how equity and diversity may be relevant, how do we want to return individualized research results to participants to motivate their behavior, and all of these things that I think are really important as you get into this area. And then, just to kind of bring it back, what we're really looking at is using now behavior to understand neuropsychiatric dysfunction and to do it both at the really fine time scale, where we can pull together data from phones and wearables and video and audio and look at, microsecond, or maybe not microsecond, millisecond level variance in people's behavior, but also then be able to zoom out at the level of developmental trajectories and follow people over thousands of days. And with that, I think moving beyond kind of more the caricature version of mental health disorders and something that could be more computationally defined and help to guide treatment planning. So with that, I just want to make one final point, which is I was part of a British Journal of Psychiatry debate a few years ago about will AI eventually replace psychiatrists? I came down on the opponent side. I don't think they will, but I do think it's relevant that lots of tools like this are out there. And I think one of the things that maybe we'll talk about in the question period is how do we know when a tool is is ready for use? And who gets to decide that? Is it just the marketplace? Startup creates it, it gets out there. Is it the government? Or is it just treating physicians or patients? Who actually is going to be the ones making these decisions? So with that, I will wrap up and just quick plug that we have a conference in Tampa in the fall if anyone's interested in data science and mental health in particular, and we welcome everybody there. But thanks for your attention.
Thank you so much. I think we have a couple questions, time for a couple questions before the break.
Oh, sorry. Great talk. Thank you. I was wondering what you think are how would you characterize the informational gains from these types of granular data, and are they worth, from the patient's perspective, the kind of privacy trade-offs?
Yeah, that's a great question. So I would say it's going to be different for every patient, but that's where, for the more applied stuff that we've been doing on the inpatient units, the informational gain that I think the doctors get from these sleep reports is more substantial than other things that we have tried so far, in the sense that they already have a way of thinking about what sleep means, as opposed to gestural expressivity or something. There's no number for that you get taught in med school, whereas for sleep, they already know about sleep, and they know that it's important to measure, but they don't measure it. And so being able to provide a report back to them. And I didn't show this, but what we found was that because we actually have a clinical QI project on the unit for this, where patients, even outside the research context, are getting these watches put on them. And we found that we had to give the doctors more of a radiology-style report where we verbally describe what the results showed, and then they can skip down to the impression, because that's what doctors do. They don't, the information gain, if you're not a cardiologist, is you read the impression. So we find that once we can distill it and we have enough familiarity to make sense of it for them, there's more buy-in, there's more understandability. But I think the question, about where location will be useful to patients or language features and other things, I think we will get there. I think when you look at enough of these, there's certainly studies where you can show patients back their own data and they can start to see things that will be helpful for them. But seeing just change versus illness-relevant change, I think will move it from being interesting to being actually clinically relevant for them. So, yeah.
In the case of the UK actigraphy study, I guess one of the main relevant things that you kind of pulled from that was that as you age, you get less active. But I have a question about so that was specifically on people from the UK. And so my first question would be how do you discern that that isn't at least partially cultural, or at least the exact slope of that change?
Sure.
And then also, how do you make the determination that that's the healthy or normal progression rather than a symptom of frailty?
Yeah. Great question. I mean, I think with any epidemiological scale data sets, you're pulling in people with and without conditions, right? So looking overall, the dots I showed you were over all of the patients in the sample without respect to whether they had, whether they exercised more or less or what kinds of conditions. But you can do all those kind of analyses because they do have a lot of longitudinal data on how they went on to do. So you can look to, try to infer causality about whether people who were at the higher end did better or worse or whatever. Because of course there are tons of counterexamples. You saw in the plot, it's a huge spread. Same would go for the question of does this generalize to other cultures, right? We don't know. It's just that UK has taken the valuable step for humanity to start collecting that data. All of Us project. There's other studies in the US that have similar size data sets. Now, I would say some of those larger studies have taken shortcuts that I think may be problematic. All of Us didn't use a raw accelerometer. They used a Fitbit, and you can't derive the exact same numbers from Fitbits because it's step counts and stuff. So hopefully, through communities like this and more recognition of where the important signals are, we can motivate larger and larger studies that capture all of the kind of cultural and illness level variance that feeds into it. But I guess that's the idea here is just how do we take population level data sets and then apply them in smaller cohorts and at least have a sense of where this person's, what this person's normal behavior is relative to another group population.
Thank you.
So at the beginning, it sounded like you were trying to shrink the time to diagnoses. Different metrics that you showed, they were obviously on different time scales. Where location, it seemed like it was months. Activity was on the order of weeks. AV interactions could be minutes or just multiple trips. I guess have you identified a sweet spot of saying that to make a diagnosis we would need at least 1 week of recordings across all metrics? Is there anything done on that scale?
Yeah, it's a great question. So we have these two cohorts. One that we follow for a really long time, year time scale, with the idea that they'll manifest their condition. Because we didn't want to assume, based on historical data, that they have bipolar disorder. We wanted to wait long enough that they have an actual manic episode, right? Because if they just have depression, then we can't diagnose them with mania, with depression, with bipolar. So in that context, we had to just wait. And the reality is it was very variable. So some people had an episode within weeks. Some people, like the guy I showed, other people didn't have an episode for 3 or 4 years. So how you would make a diagnosis in that context, now would the face data be enough to show that this person had a bipolar disorder? That's what the other study was for, where we follow people on the inpatient unit, where they're coming in already manic or already, and there we're just following their trajectory back to recovery, but it happens over a much shorter time scale. So the shorter answer is I think if you can follow people during changes, you have a much better chance of diagnosing them, and so when people are in the inpatient unit, they're changing on the scale of days, and you have much closer tabs on exactly what they're getting, because it's not just what they're manifesting, but it's what in response to what, right? So if I give somebody lithium and their mania goes away, then okay, well, they had bipolar, whereas on the outpatient, it's much harder. So I don't think there's a simple answer, but clearly you need change to occur, and you have to be measuring the right things when that change occurs, and having full handle on both the ins and the outs, right? What are they getting and what are they manifesting will be the best way of doing that. But how we've the optimal way of instrumenting that in a either inpatient or outpatient setting, I think will be very dependent on the person. And I think the dynamics of change and episodes is different depending on what conditions. So, but yeah, it's a great point.
But that test you showed, it just looked like a much better PHQ-9. That looked like that's where it's heading.
Yeah, well, I think, right, and being able to go beyond just depression, right? Trying to think transdiagnostically and something that is naturalistic, but also something that, you could do maybe in a self-administered way. So.